Today I'll talk about immunotherapy in cancer. Immunotherapy is a type of cancer treatment that boosts your body's natural immune system to find and destroy cancer cells. Unlike chemotherapy, which directly attacks tumors and some healthy cells, immunotherapy trains, equips the immune system and immune cells to recognize and fight the cancer. So, why do we need it? So, normally, your immune system patrols your body to destroy harmful or abnormal cells. But cancer cells are tricky. They can hide from the immune cells and secrete certain signals to make the immune system feel that everything's okay. So, how does immunotherapy work? What are the mechanisms behind it? One, immune checkpoint inhibitors. So, cancer cells use certain proteins, checkpoints, like a shield to tell immune cells to stand down, everything is okay. Checkpoint inhibitors act like scissors, cutting that shield so your immune cells can see and attack the cancer. 2. CAR T cell therapy. Doctors can extract some of your immune cells, alter their genetics in a lab so they're perfectly designed to seek out your specific cancer, multiply them, and put them back into your bloodstream to hunt the tumor. It's like training an immune cell outside the body and putting it back inside the body. 3. Monoclonal antibodies. These are lab-made proteins designed to attach to cancer cells. Once attached, they can flag the cancer cells so immune cells can easily recognize and destroy them. 4. Cancer vaccines. Just like a flu shot teaches your body how to fight a virus, these help your immune system learn to recognize cancer proteins as a threat. Some things to know about immunotherapy are that it has long lasting effects because it teaches your immune system. Your body can remember the cancer cells, offering protection that lasts even after the treatment ends. 2. It's a targeted approach. It generally damages fewer healthy cells compared to other therapies, leading to milder side effects. But one disadvantage is that it's not for everyone. Immunotherapy is a major breakthrough, but it does not work for every cancer type or every patient. Why can the body's immune system recognize pathogens but cannot recognize cancer cells? So, whenever bacteria and virus enter our body, they have these proteins on their cell surface called antigens, which mark them as non-self. So, the body's white blood cells are checking the body continuously for this non-self marker, and when they see this on bacteria or viruses, they can trigger an aggressive immune response. But cancer cells develop from normal healthy cells. So they don't have this non-self marker on their cell surface, and therefore they can disguise themselves as normal cells and fool the immune system. So how do cancer cells fool an immune cell into thinking that they're normal cells? So normal cells have something called PDL1, programmed death ligand 1, on their cell surface, and immune cells have PD1 on their cell surface. PDL1 of a normal cell binds to PD1 of an immune cell. And this binding means that everything is normal, the cell in front of you is a healthy cell, and an immune response is not required. So this is how an immune cell checks for any virus, bacteria, pathogen, etc. The problem is that cancer also has PDL1. So when an immune cell is checking for pathogens or foreign cells on the basis of PDL1, cancer also expresses PDL1, and now the PD1 of an immune cell will go and bind to PDL1 of a cancer cell just like it did in the normal cell scenario. And this results in stopping the production of TNF alpha, IL-2, IFN gamma, which results in stopping the immune response that is required, because this is a fake handshake which the cancer does to show the immune cell that it is also a normal cell. So therefore, in immune therapy, what is done is either the PD1 receptor is blocked on an immune cell or the PDL1 receptor is blocked on a cancer cell, so that this fake handshake cannot occur. Now the problem is, if PD1 is blocked on an immune cell will not be able to check the normal cells also. It will not be able to do the real handshake it has to do with normal cells as well. Therefore, PDL1 blocking is a better idea. Now, I said that normal cells have PDL1. So if I block PD-L1, that means that the immune system will flag normal cells as foreign particles or cancer cells maybe. So the backup mechanism here is that normal cells also have PD-L2, which is a backup receptor that can bind to PD-1 and do a similar kind of binding handshake, which results in stopping the immune response. I've been talking about immunotherapy. Today I'll talk about the relation between melanoma, which is skin cancer, immunotherapy, and vitiligo. So, some melanoma patients develop vitiligo during immunotherapy. These patients show better treatment response and better survival. This happens because immune cells that are attacking melanoma also attack normal cells. So why does vitiligo happen? This is like friendly fire. So immunotherapy activates immune cells to attack melanoma cells, that are skin cancer cells. Melanoma cells and healthy cells share similar targets on their cell surface like GP100, MART1, tyrosinase. So that's why immune cells accidentally attack healthy cells also, and this causes the white patches called vitiligo. The condition vitiligo is an autoimmune disease where the immune cells attack melanocytes. People with vitiligo may have lower melanoma risk because their immune system already targets pigment cells, it's already hyperactive. Immunotherapy induced vitiligo happens in melanoma patients receiving immunotherapy, and the immune system becomes highly activated and attacks both melanoma cells and normal cells. This is called melanoma associated leukoderma or vitiligo-like depigmentation. So the takeaway is that vitiligo during melanoma immunotherapy acts like a visible sign that the treatment is working, and it often suggests a strong immune activation and better outcomes. I'll talk about a very specialized and fascinating type of immunotherapy called CAR T cell therapy. So I said two things before. One, cancer heterogeneity. Every cancer is different and on different levels: genetic heterogeneity, epigenetic heterogeneity, microenvironmental heterogeneity. And the second thing I said is that the cancer knows how to hide and run away from the immune system. CAR T cell therapy can overcome both of these things. Let me tell you how. So, how does CAR T cell therapy work? First, blood is taken from the patient and a machine will separate the T cells, the white blood cells, from this blood. Then these cells are genetically modified. A gene is added in these cells, and that gene expresses CAR, chimeric antigen receptors. Then these cells are grown, multiplied. Then the patient is given chemotherapy first to prepare the patient for CAR T cell therapy, and then these millions of cells with the CAR, chimeric antigen receptors, are added to the patient. Then the CAR T cells act like radars or sensors to find cancer cells. Then they bind, attack and destroy cancer cells. They can multiply further inside the body and stay active for long-term protection. The cancers that are treated using CAR T cell therapy as of now are leukemia, lymphoma and myeloma. But there are certain side effects of CAR T cell therapy. They are: 1. Cytokine release syndrome, which means a strong immune reaction, fever, flu-like symptoms and low blood pressure. Second, neurological problems, confusion, difficulty speaking and tremors. This is an important topic and this shall be continued tomorrow. What is so special about CAR? How does it recognize cancer cells? So a normal T cell needs major histocompatibility complex molecules to present cancer proteins, and only then can a normal T cell recognize a cancer cell and do anything about it. But a CAR T cell does not require MHC. So a CAR T cell has single chain variable fragments, SCFVs, that can just directly grab cancer. It does not need MHC to present cancer. After a single chain variable fragment grabs the cancer, the inside of a CAR T cell is activated There's a protein called CD3 on T cells which helps activate T cells whenever danger is detected, and CD3 zeta is a fragment of that protein CD3, a part of that protein, which is like the ignition switch, the activation switch. So CD3 zeta is the attack switch, attack signal. CD28 is an energy booster. So it gives extra activation and energy, and it makes the T cells stronger, helps rapid multiplication, improves survival, and makes the response faster. So CD28 is like a fast energy booster. And then there's 41BB, also called CD137. So this is like a long lasting battery. It helps T cells survive longer, it prevents exhaustion, it improves long-term persistence, and it keeps cells active for months or years. So 41BB is like the survival battery of a cell. So after the CAR T cell uses CD3 zeta for attack, CD28 for energy, and 41BB for long-term endurance, what happens is it releases toxic proteins, perforins, that create holes in the cancer cell membrane. And then granzymes enter through these holes and force the cancer cell to self-destruct, apoptosis. So there are 3 generations of CAR T cells. First generation is only CD3 zeta signal. So it can recognize a cancer cell, but it will become weak very quickly. Second generation has CD3 zeta and has CD28 or 41BB, one of them, one of the boosters. So T cells can survive longer and multiply. Third generation has CD3 zeta plus both the boosters, so designed to be stronger and long lasting.